Welcome, everyone, to Frontiers of Brain Health. This is our first session of 2021, and oh, what a beginning it is. Today we have 1,200 people signed up. My name is Dr. Sandy Chapman. I am the Chief Director of the Center for Brain Health at the University of Texas at Dallas and a professor in the School of Behavior and Brain Science. I cannot even tell you why brain health is such an important endeavor for all people, not just a small percentage, but all of us. And I think the pandemic and all of the crises that have stacked has risen to make us realize why that is the case. Literally in 10 days, we are going to be part of the Organization of Economic Cooperation and Development that reimagines economic reimagination of how we can restart our economy with ambassadors all over the world and two people that are very integral to our brain health project. So you might want to tune into that as well. Now on to Dr. John Cryan. Dr. Cryan, wow, he has more diverse professional name specialties than I've ever seen. He's a neuropharmacologist, microbiome expert, neuromicrobioticist, a behavioral neurogeneticist, a brain health expert, and so much more. I always say our health starts and ends with our brain. John says, Dr. Cryan argues that health starts in our gut. Maybe we're both correct. Our brain is in our head and it's integrally wired to the digestive system as they interact wildly. We have so many questions. I would like for you, as you're listening to this, to type your questions into the Q&A, and we'll see how many we get to, because I have a lot too, John, so get ready. 2 years ago, when we invited John to be part of us and he came, everyone had so many questions, and at dinner, literally, he had everyone eating out of his hand, picking his brain, and watching what he was eating. There is probably not very many people that have reached the stellar career in such a short time as Dr. John Cryan. I don't need to read you his bio because you got much of that ahead of time. But last year, he had an H index of 91. This year, it's 124. A couple years ago, he had 440 publications. Now it's 500. He has been published in every single prestigious journal there is: PNAS, Neuron, Nature Reviews, Neuroscience, Molecular Psychiatry, and the Journal of Neuroscience. He is also the senior editor of Neuropharmacology, of Neurobiology of Stress, and Nutritional Neuroscience. And he's on the editorial board of 15 journals. Plus, if that's not enough, he's edited 4 books. He was elected a member of the Royal Irish Academy just a few years ago. In his spare time last year, he was president of the European Behavioral Pharmacology Society. Whew. How can you have a healthy gut, John, and still achieve at this level? I want you to know that we are so honored to have Dr. John Cryan as one of our esteemed collaborators on the International Brain Health Project. You'll see why we are honored to have him guide people how to optimize their brain-gut health. Please join me in welcoming Dr. John Cryan. Welcome, John.
Thank you so much, Sandy. What a real pleasure it is to be here, and thanks so much for those very kind and generous words. I'm delighted to be associated by the Center with the Center for Brain Health. The audacity of what you're trying to achieve and the importance of what you're trying to achieve there in Dallas is just amazing, and I'm always in awe of you and your whole team there. And I would really encourage everyone who's listening to get involved in any way they can with the research that's ongoing. It's so important. Our brain health is so important to all aspects of our health. That said, as you alluded to, today I want to also maybe get you to think a little bit about your gut feelings or your gut instincts. I don't want you to be too gutted, but we may make a few gutsy moves or gutsy insinuations in my talk. So let's see, hopefully over the next 35, 40 minutes, I can encourage you to think a little bit more about what's going on in your gut. Now, this is a famous painting by Matisse, and it hung in the Museum of Modern Art in New York as part of a retrospective exhibition in 1961 like this. It's called Le Bateau. And but it wasn't until a very eagle-eyed stockbroker called Genevieve Hebert looked at it and said, there's something wrong here. You know, something is wrong. And people have been looking at it for many weeks, including Matisse's son. And what was wrong was they'd been looking at it the wrong way around. It was turned upside down. And so hopefully I can convince you that perhaps to look at things the right way around, we may have to just turn them upside down and look at them in a different perspective to see them in their proper way. And that's kind of one of the take-home messages that I have. Now, I work, like probably many of you on this call, I work in a medical school, and in medicine, everything more or less goes back to Hippocrates. And Hippocrates is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. Now, when I talk to my neurology or psychiatry friends, they would look at me quite strangely if I start saying that. But hopefully, I can also convince you that there may be something in this. And like one of the main things that I do want you to take home from my talk today is to remember that we are actually living in a microbial world. That is, the microbes were here long, long before humans. And so this is really one of the things that informs a lot of my thinking. If you start putting things in an evolutionary context, you start looking at it a little bit differently. And I want to give you some facts about these microbes that we have. If you think of in terms of the sheer genes that we have in and on us, we have basically, we're more than 99% microbial. Now that's very humbling to think about. All of the money we spent on the Human Genome Project, and it's for less than 1% of our genes. The weight of our gut microbes is roughly about the same as our brain. So as a neuroscientist, I find that also quite humbling. It's also thought that we have more microbial cells than we do human cells. It was thought to be 10 times, it's now been revised down to about 1.3. So we're still more microbial, but just about. So just think the next time that you might go to the bathroom and get rid of some of these microbes, that you're becoming more human, and what that might mean. Now, what my lab is really interested in is trying to really understand the relationship between these microbes and the host and the brain in particular at key windows across the lifespan. In early life, during that perinatal period, in adolescence, all the way up until aging. And I'm just going to touch on each of these in my talk today, just to give you a flavor of the type of work we do, which is really work based on the translational interface. Now, where does it all begin? Well, for the most part, we're taught to be sterile in utero, and we get our microbes as a birthday present from our moms as we emerge through the birth canal. And these microbes start then to inform the developing immune system and help programming many systems within the body. And there are a variety of factors that can influence the composition of these microbes, especially in early life. Among them is the mode of nutritional provision, whether it's breastfeeding or bottle fed. Host genetics and epigenetics does play a big role. The environment we live in, exposure to antibiotics, all will play an effect on this. And one of the things we've been interested in particular, is mode of delivery at birth, because we know that this if you interfere with the actual mode of delivery, then you interfere with this kind of evolutionary handing over of the microbes. So it's like the baton race doesn't happen. You drop the baton because, and this goes back to really classical work from Gloria Domingos-Bella's group, now at Rutgers, where she showed in this paper that infants born by C-section have a very different microbiome than those born per vagina. And this has been studied in many, many cohort studies, the largest being the TEDdy study, which is over 12,000 individuals, showing that birth mode changes the microbiome. And as Nature put it here, it changes this kind of security blanket that is so important for this developing immune system, so important for developing all of the systems in our body. And work from Trevor Lawley's group more recently showed that it's not only that you're missing microbes, which is part of the problem, but it's also the blooming of some pathogenic microbes overall. And so we've been interested because we know that C-section from epidemiology studies is associated with an increased relative risk of many immune-related disorders like asthma and allergy and type 1 diabetes. But we know very little about it in the context of brain health. And that's something that we've been very interested in studying. First of all, in animal models, we showed just in the paper out this year that animals that are born by C-section have enduring behavioral effects, especially in social domains, and that we could reverse many of these effects by specific bacteria, in particular bifidobacteria. But this is mice, and I think most of you are interested more in humans. And so what we wanted to do was, could we reproduce these changes in a human population? So to do that, we took, I was going to say normal, but Irish medical students, and stratified them based on their mode of delivery. And then we needed a way to see how they respond to stress. And we have very good ethical ways of acutely stressing people in the laboratory. They come in and they are asked to pitch their dream job in front of these very grumpy looking people in white coats, and then they're told to do a mental arithmetic task. And in doing so, then we can mount a very good psychological and biological stress response. And what we found was individuals born by C-section had this increased psychological and immunological response. But that's acute stress. What about chronic stress? So how do we chronically stress in an ethical way people? Well, one of the good things about being an educator is we do this all the time by subjecting our students to exams. And so we can look at the long-term effects of exposure to these chronic exam periods in these individuals. And again, on a biological and using Cohen's perceived stress scale here, we were able to show that there is an increased perceived stress in individuals that were born by C-section, in individuals that had their microbiome changed. Now, there are many other factors at play. It may not just be the microbiome, but our animal studies show us that we're able to reverse many of these same type of deficits by putting the microbes back early on. So it offers us some opportunities, especially as we see C-section rates very high in many parts of the developing and developed world. And so this is quite interesting. But it's probably not enough for my neuroscience colleagues. My neuroscience colleagues are a skeptical bunch, and they want more evidence that the microbiome in early life is important for brain and behavior. And so how do we get more evidence? Well, probably the best way to get evidence is we learn from engineering. We take something out and see what happens. And so the concept of growing up germ-free without microbes isn't new at all. It goes back to Pasteur's time, but it allows us to answer that question: is the microbiome involved, yes or no? And that's something that we've been working in our mouse models over many years now. And we found some amazing findings in these mice. First of all, one of the most surprising was we found that myelination. Now, myelin is the insulation to allow appropriate nerve conductance. And we found myelination, and particularly in the prefrontal cortex, in that really important area of the brain for higher cognitive function and the top-down regulation of the stress response, that there was a hypermyelination in these animals without bacteria. Adult hippocampal neurogenesis. The hippocampus is the area of the brain involved in learning and memory. And the birth of new neurons, neurogenesis in this hippocampal area, occurs throughout our entire life and starts to fade as we age. And to keep that going by environmental stimulation like exercise and enrichment, but also through antidepressant medications have been shown to increase it as well. And we're very interested in the processes of this. And so we found in these germ-free mice, again, that the neurogenesis regulation has gone completely out of kilter. We find changes in a variety of neurotransmitters and changes in synaptic plasticity. I'll show you some of that new data we have on that. Some changes in dendritic growth. Microglia of the brain's immune cells, work from Marco Prince's group and Sonia Gorel have shown very nicely, and we're working on this as well, that there is a change in the activation state of microglia in these germ-free animals, and of course the blood-brain barrier, this key protective barrier that mines our brain in so many ways. Work from Sven Pettersson's group and Karolinska some years ago has shown very nicely that this is dependent on having microbes in the gut. So here you go. This is a neuron from the amygdala, the region in the brain involved in fear, involved in pain processing, involved in social behavior. And this is a basolateral amygdala neuron, and you can see that it's arborized really, really heavily. And this arborization is very much like what we see in chronic stress states. This is really hot off the press data just in press now, where we showed that long-term potentiation, LTP, the molecular substrates for memory formation, that in male, but not female animals, and that's a theme we keep coming back to over and over again, these sex differences. In males, we see a clear deficit in the hippocampus, in this electrophysiological correlate of learning and memory. And so they're not able to mount a proper LTP response, these germ-free male animals, but the females can. So that's interesting. What does this translate to at a behavioral level? Well, it translates to deficits in amygdala-dependent fear recall, changes in visceral pain, And one of the things we've been really interested in is social behavior, because all of the effects we see seem to be much more prevalent in males, and many of the deficits of social behavior are also more prevalent in males. And so mice, like humans, are quite social. So if you give them the chance to spend time with another mouse or an empty chamber, they'll gravitate towards the mouse, but not if they're germ-free. Mice, well, perhaps like some humans, can be a bit fickle. So if you give them the chance to spend time with a new playmate versus their everyday playmate, they'll gravitate towards a new playmate. But again, not if they're germ-free. And so that really was really exciting data for us to really drill into the mechanisms, and I won't go into the details. We started in looking at when animals are in social interaction, what happens to their brain, molecular pathways and genetic pathways. And we find, for example, a real enrichment in these germ-free animals in splicing, RNA splicing. And of note, work from Dan Geschwin's lab is showing alterations in RNA editing in individuals with postmortem tissue from individuals with autism. So there seems to be some convergence here about social brain and its relation to psychopathology and the microbiome. And, you know, this led us to start riffing on this some years ago. Roman Stilling was a postdoc in the lab, and we started thinking about it, and we started thinking, well, what does it mean from an evolutionary perspective? Why do we need microbes in our gut for normal social interactions? And you know, we started thinking, well, what's in it for the microbes? And we started putting some of these ideas together. And then we collaborated with Seth Bordenstein, a wonderful evolutionary microbiologist in Vanderbilt. And we put some of these ideas to Seth, and he basically told me I was completely wrong, even wrong to even ask him that question. And that's why it's so great to have such wonderful people who are coming at this topic from different fields. And the reason I'm wrong is that I've forgotten something that I've already told you guys. And that is that the microbes were there first. And so that we must contextualize all of brain evolution. The brain has never existed without signals coming from the microbes. We have co-evolved. They're our friends with benefits, but our friends perhaps with social benefits. And that's been quite an interesting journey for us to start thinking about the brain in that way. And in this paper that we had out in Science last year, we wanted to really look at this across the animal kingdom. And when you start looking at when you change social hierarchies and social dynamics, whether it's in insects all the way to non-human primates, you interfere with the microbiome. And likewise, if you start interfering with the microbiome, you interfere with social structures and social dynamics in a variety of different ways. And that's really, really intriguing to me, especially when you think about what's happening right now during the pandemic, when we're altering our social structures so much. When I would love to be in Dallas giving this talk in person and having a really nice social interactions with Sandy and her team, but here I'm not. So we are actually having very, only time will tell what's this doing to our microbiome, what's this hypercleanliness doing to our microbiome, and whether that has any long-term effects on how our microbiome signals to our brain. And what we're beginning to understand from a social construct then is that we have a microbiome gut social brain axis. That there's something about the circuits of the social brain that make it somewhat wired to receive signals from microbes. And we know that a variety of factors in the environment, we talked about mode of delivery already, genetics, the sex differences, the diet, we'll come back to. I spend a lot of time focusing on medication, the environment we live in, whether we're in a city or in a green area, and the social interactions that we have will all influence this. And then the questions that we have, and this is something that I'm working with colleagues in France and Italy and Germany on, and is trying to really look at these, can we modify some of these influencing factors to influence how the microbes talk to the brain to maybe change how we behave under normal circumstances? And this is quite provocative and a little bit out there, but it remains, can we change how we cooperate, our teamwork, our empathy, our trust, all of our social decision making? Can we change these by changing what's going on in our microbiome? We don't know yet. This is a really exciting area, and a lot more research is needed in this, but it's something that we're very excited about overall. And then to conclude on the early life bit, we really beginning to see that while the study of critical windows in neuroscience is really well developed, and we understand there are these windows for sensory and cognitive development, and that when these get disrupted, it can lead to certain brain disorders. We also begin to realize that there are critical windows in our microbiome. And we need to start juxtaposing these together and understanding these relationships at these key windows as we mature and go forward. Moreover, the well-known Barker hypothesis of the developmental origins of health and disease. We can't neglect that also, even in the prenatal environment, even though we may be largely sterile, the mom's microbiome is going to get altered and can have the possibility to change through metabolites, can change what's going on in the offspring. And so we're beginning to see that if Barker talked about different pillars of the developmental origins of disease, but maybe for brain health and disease, we need to add the microbiome as a fourth pillar. In addition to the prenatal, the postnatal, and host genetics, we also need to think about the microbiome, because the microbes were there first, and we can't neglect the first founding fathers, if you want. So let's move to adolescence. Adolescence is really, really important area. It's probably one of the hottest areas in biological psychiatry and psychology right now, because we're really dealing with a very different brain. And I'm professor of anatomy here. And any of you with teenage kids or understand the teenage brain will realize that the anatomy of the teenage brain is very different. But most importantly, we're realizing that the teenage brain is undergoing a huge amount of remodeling, and is a very important, critical time period, and one which is very much under construction. Also, what we're considering adolescence is now being extended well into the college years, and we understand that there's still a lot of synaptic pruning, and understanding this relationship between, for example, microglia and neurons, and how the pruning that's undergoing at this window is really, really important. And because of the emergence of a variety of brain disorders at this time window. And so a lot of focus in the adolescent psychiatry field is focused on insults like stress, alcohol, poor nutrition, drugs of abuse, inadequate sleep, and what they may have on the brain. But also we need to study what they're also having on the microbiome. And so that's really important overall. And so whether we're talking about it in the context of disorders like adolescent onset schizophrenia, ADHD, eating disorders, mood disorders, anxiety, we need to really think about this. And this is something we've been working on. I won't go into the details in the interest of time, looking at animal models, modifying diet, looking at the effects of stress, and looking at the effects of antibiotics in at this critical time window. And that's really, really good to be start to detail some of the mechanisms. We need to now move into translational studies in humans to be able to see how much this stands up overall. And then I'm just going to move to aging, because we're very excited about this because the fundamentals of healthy aging may actually lie in the gut. And this goes back to classical work from my colleagues here in Cork, from Paul O'Toole, which showed in the ElderMet study that the diversity of the microbiome starts to decrease as we age. Now, in a simplified fashion, diversity, like in all aspects of life, diversity is really key. And we want to maintain diversity as much as possible. So I really want to see, how we move forward with this overall. So in some studies we did a few years ago, we showed that animals that were aging, that they had increased diversity overall in their the changes in their diversity in their microbiome. And that led us to start thinking, could we change this? Is there anything we can do about what's going on in the microbiome in these animals? And so what we did was we treated them with a diet that was rich in inulin. So we wanted to see, could we use a diet to actually stop the effects of this aging? And with aging, and for this studies, what I decided to focus on was middle age, for obvious reasons. It's a time window I'm very interested in right now myself. And so in these middle-aged animals, there's neuroinflammation going on in their brain, and we were able to treat them with a diet rich in inulin. Now, inulin is a prebiotic that's present in a lot of vegetables, like leeks and artichokes. And so in the animals that have been given the inulin, you can see here in green, I won't go into the details, but we're able to attenuate the age-induced neuroinflammation. So that's remarkable. So, what this means is, and this is work from Marcus Boehm, a postdoc in the lab, together with my colleagues Ted Dinan and Harry Shellikens. What it really means is, if you're thinking of having a midlife crisis, maybe forget about the motorbikes and turn to the leeks instead, and that they may give you a better overall effect. Where we're going in this field is really, and where we need to go, is to try and understand how on earth is any of this happening? How can microbes in the lumen of our gut send signals to our brain? And we're really just at the beginning of trying to figure all of this out. And then can we translate any of that to the human condition? One of the ways is through the vagus nerve. The vagus nerve is this long wandering nerve that receives signals, and it's bidirectional. And we've been very interested in this for many, many years. And what we showed almost a decade ago now, together with John Bienenstock's group in Canada, we showed that when we treated animals with a certain bacteria, they had lactobacillus, they had reduced stress response, and they had really, really marked effects on brain. But when we cut the vagus nerve, all of these effects were gone. So what this means is that what happens in vagus doesn't just stay in Vegas, but will actually affect our emotions. Now, that's my oldest joke, and those of you who heard me talk have heard it before, but I was really proud to get it into this Neuron article last year so that we could actually say it. But it tells us how important this is, and we know that vagal nerve stimulation is really important for lots of different disorders, and maybe the microbes are hijacking this neural pathway to get into the brainstem and then stimulate emotional circuits and limbic circuits thereafter. So that's been quite an exciting adventure to try and look at this, and we're digging into that more mechanistically. Also worth remembering is that the microbes are like little factories producing all kinds of weird and wonderful chemicals that our bodies wouldn't have without them. And so we thought, well, maybe we'll just bypass the microbes altogether. Let's go to the end products. So one of these end products, for example, is short chain fatty acids. And so these are like butyrate, acetate, and propionate. And so again, this is an animal study, we decided to just give animals these short chain fatty acids, and we're able to show that it blocked the effects of stress. both at the gut level and at some of the behavioral levels. So that's quite encouraging. And people now are looking at trying to encapsulate these and deliver these in humans to block some of the effects of stress as well. And a lot of innovation going on in this field. So it's not just the microbes that are there, but it's the little chemicals that they're producing, their metabolites. And the field is really looking at these metabolomic approaches more and more now, because they give us some mechanistic insight into how they're affecting the brain and the body. Sandy already mentioned, and I don't have to remind you about this, but I'm putting in here just to remind myself to talk about stress, because that's what I'm really interested in doing, is understanding the effects of stress in our brain and our body. And we're all going through different types of stress. People talk about us all being in the same boat. Well, we're not in the same boat. We're in the same ocean, but we all have our own different boats, and them boats are dealing with stresses in different ways. And how the brain gut axis is dealing with that is going to be really important. And what I want to reinforce about stress is that stress, and I'm talking mainly about these types of chronic stress or traumatic stress, is a whole body syndrome. It doesn't just affect a few neurons within the dentate gyrus of the hippocampus, and we love those neurons. We study those neurons, they're fabulous, but it affects our immune system and how it talks to the brain. It affects locally in our gut barrier function, and drives a pro-inflammatory phenotype. So we've been really focused on this. In a lot of our animal studies, we've shown that stress will have changed the microbiome. And more recently, Thomas Bastianson is a bioinformatician in my team, and he's really interested in longitudinal measures of stress in the microbiome. And we came up with this idea of volatility. And basically, the more stressed you are, the more volatility there is in your microbiome, the bigger the distance of movement in your microbiome. And we show this first in animal studies that were going social defeat stress, but also back to our students that get exams. We're able to show a very clear relationship between their perceived stress score and this volatility, and also in relation to their hypothalamic pituitary adrenal axis function, in relation to their cortisol awakening response and the volatility as well. So it gives us an idea of another measure we have within our microbiome to assay how stressed we are, and maybe look at ways to alleviate that by targeting the microbiome. And that brings me to the concept of psychobiotics. This is a term we coined here in Cork, my colleague Ted Dinan and myself, some years ago. And really what it means is targeting the microbiome for mental health benefits. There are different ways of doing that. You can do it through probiotics. I already mentioned some prebiotics, which are dietary substances, synbiotics, which are combinations of both. Parabiotics, they don't even have to be alive. This is where dead bacteria are able to induce effects because of what's decorating their cell wall. Or postbiotics, like the short chain fatty acids that I talked about. The other one is the fermented foods, which I'm really excited about as well. And we've been working a lot on together with my colleagues here, Paul Cotter in Teagasc and overall. And we've written this book for the general public to try and encourage people to start thinking about their gut health when we're talking about their brain health. And we call the book was titled A Revolution. Well, it's really the beginning of a new revolution, a new way of thinking, a new way of turning things upside down. And that's the kind of journey that we want to be on. We have lots of data in animals. I won't delay on it too much, just to show that across all of these different modalities, we've been able to show that they're able to modulate behavior very, very nicely. But, so many brain disorders have been solved in animals. We need to do human translation. And that's why I'm really pleased to be associated with the Center for Brain Health. And so one of the things we wanted to do was what if we took one of these bacteria, this biflongum that we had worked on in animals, and put it in a group of healthy volunteers that we bring into the lab, that we subject to an acute stressor. In this case, we used what's called a socially evaluated cold pressure test, where you basically put your arm into an ice bucket of water and you mount the stress, and there's someone watching you in a white coat taking notes about how brave you are and how good you are. And so there's a good psychological component to it. And we're doing brain EEG. This is Andrew Allen, the psychologist on the team who was doing the EEG. So we're measuring what was going on. And what we found was that the individuals that had taken the biflongum, they didn't mount the same stress response. Remarkable, really, really encouraging for me as a basic scientist to see something coming from our animal studies go all the way to humans. Paul Eng's group in Tübingen took the same bacteria. We advised them about what bacteria to take. They then put people in the scanner, and they looked at them using a virtual reality cyberball task, where they're playing a ball game with two others in the scanner, and all of a sudden the two other people start ignoring the person in the scanner. And your brain doesn't like to be ignored. And so it mounts a stress response. However, those that had taken the biflongum didn't have the same stress response. Remarkable. Small study, but really remarkable. Healthy people, that we could actually mount this. So this is really encouraging about the whole concept of the psychobiotic. Then we took our lactobacillus, which is our most robust probiotic in animals, and we looked at it in some of the same paradigms in humans, and it did nothing. You cannot get more negative data into the one paper if you tried. And so this highlights the limitations of this of translational neuroscience research. We can't translate everything from animals all the way to humans, and that's just part of what we do and how we do it. But it's not specific to the microbiome field. It's in all aspects of biological psychology in general. More recently, we've been looking at a fiber, because fiber is basically the thing that really your gut microbiome loves. And it ferments this fiber into these short-chain fatty acids and other key chemicals. And so we looked at this was a polydextrose, and we found some really interesting cognitive effects of this fiber using the Cantab battery of cognition, and we just published this recently. And we measured the microbiome in this, so we're able to see how much the microbiome could be playing in terms of this. But it's still surprising how little we know about fiber and cognitive function. And fiber is cheap and it's available. So it's an interesting aspect, and it's something we're going to be studying once we can get back into the lab to do more of these studies after the pandemic. In relation to mental disorders, we've been very interested in the role of the microbiome in major depressive disorder, and really beginning to start to put some of these pieces of the puzzle together, buoyed on by big studies like this study from Joran Raas's group in Belgium, showing that there's a very distinct microbiome composition in individuals who are depressed, and they were able to replicate that in another large cohort of individuals in the Netherlands, as well as in treatment-resistant patients. And we're beginning to see that depression is really a trinity, and this is my Irishness coming out. I like to use this trinity logo or emblem to say that we have disrupted inflammation, disrupted stress, and microbiome really coming together, giving rise to depression. And these mechanisms are how they interact with each other are very important, and that any successful treatment then should be able to target all. Going back to looking at plasticity in the brain, we're beginning to see then how factors like diet and exercise and a healthy immune system work with the microbiome to really improve and enhance plasticity and neurogenesis, whereas other factors on the other side start to knock it down. And so only time will tell how much we can target the microbiome to improve mood. But it's not just in psychiatry. In neurology, is really waking up to the whole area of microbiome brain gut axis. Of course, epilepsy has been using diet as an approach for a long time, but we're seeing this more and more in Alzheimer's disease, Parkinson's disease. I already mentioned autism, but even ALS, stroke, Huntington's disease are all now studying the microbiome, which would have been unheard of 10 years ago. And really, we're really trying to understand the relationship between diet and the microbiome. And we know from cross-cultural studies, people have gone out and looked at what the microbiome is like in ancient ancestral individuals, like in Tanzania, the hunter-gatherers, and they've shown that there's really diverse microbiome, lots of fiber in the diet. We can see then that agricultural practices have come on board, and they've started to call the diminution of the microbial diversity. And then in our Western world, and it's not just in the US, it's in Ireland, it's everywhere, we see with the introduction of processed food, overuse of antibiotics, especially in the food chain, stressful lives we live, there is an extinction of these microbes that our ancestors had. In these societies, we know that there is very little inflammatory disorders like inflammatory bowel disease or multiple sclerosis. In our modern, we know less about brain health in these populations, but it would be really interesting to see. And so the question is have we caused long-term damage by damaging our microbiome? And is there anything we can do about it? And the whole area of nutritional psychiatry really is emerging. And this is something we've been working on a lot here in Europe. This is the My New Gut project that we've been involved in here in Cork. And the ECMP, we have had a nutritional psychiatry task force to really look at how can we use diet to target the microbiome, to target the brain. and work that we're doing ongoing in the lab, a really exciting work from a nutritionist in my team, Kirsten Birding-Harold. We're really trying to triangulate the relationship, because we know diet can have direct effects on the brain for sure, but how much of the effects, the positive effects of diet on the brain are driven through its effects on the microbiome. And so we've developed a psychobiotic diet, rich in fiber, rich in fermented foods, with a very Mediterranean focus to it. And we've put it through people who have a poor diet and are susceptible to stress. A lot of them were our students, and we just did it for 1 month. And in that 1 month, there was remarkable findings at the psychological and biological level, and we're getting that ready for publication soon. And so that's really an exciting area, and it offers a lot of hope over all this, because our state of gut will affect what's going on in our brain. We have to be careful about the hype in this whole field. I don't know where we are in the hype cycle. Jonathan Eisen gives the Overselling the Microbiome Award. It's not an award I want to get among the ones I have. We have all these challenges about rodents versus humans. Correlation does not mean causation, and the microbiome field is the worst for this. For example, you can correlate the divorce rate in the state of Maine with the per capita consumption of margarine. Now, I'm Irish and we like butter, and you can come up with an idea that if someone kept coming home with margarine instead of butter, they could start fighting and it could lead to divorce. And that some of the stories that are being constructed in the microbiome field are a bit like that. And so we need a lot of caution on that, a lot of small underpowered studies. We're getting better. The lack of dietary information, that's getting better. Good psychiatric and psychological phenotyping is really required. Not everyone can do this. And we need a lot more rigorous trials and really get at mechanism. And we have issues to try and understand, because we don't know what a normal microbiome is. You can go to your doctor and get your blood pressure checked, and he can say where you are in a range. You can get your cholesterol checked, and you know where you are. But your microbiome, it's yours. It's your tree trunk of everything that's happened to you all of your life. And so unless you know what your microbiome is like when you're healthy, then it's hard to know what it's like when it's ill. And so that's where these longitudinal measurings are going to be really important. And that's why some of the programs that the Dallas team are doing is so important, to try and look at, can we use interventions and can we bolster the microbiome over time and see what's going on. It'll lead to personalized diets and interventions. We'll get a lot of understanding about what's going on in relation to host genetics. We need to be careful, so careful. My advice to anyone is, always ask, what is the evidence for this product? There's a lot of snake oil out here. There's a lot of problems in this field. So be very careful. The media also can misinterpret some things in this field very much, and you need to be really careful about how it is. But the real good news is that, unlike our genomes, which all we can do is blame our parents and our grandparents, our microbiomes are potentially modifiable. And that gives the individual great agency over their own health and over their own brain health. And so if we move forward towards a psychobiotic life, we know we can increase the intake of certain dietary substances like prebiotics that I mentioned, omega-3 fatty acids, the polyphenols that are involved in nuts and seeds, they're in red wine and chocolate, but also grape juice and onions. The fermented foods I mentioned, like the 3Ks of kimchi, kefir, and kombucha, but also yogurt and sauerkraut. And we should decrease the intake of processed food, emulsifiers, sweeteners, all things that are known to have negative effects on our microbiome. Aerobic exercise is really good on our microbiome. We should try and avoid stress, not easy. Sleep, we're really working on the circadian sleep interface in the microbiome, and it's so exciting and so much going on here. And since I've stopped traveling, it's wonderful for my microbiome because I don't get jet lag anymore. And avoiding C-section, we mentioned, supporting breastfeeding, minimize antibiotic use. And then the one thing that my wife and kids convinced me about some years ago was to having a pet, especially a dog, is really good for your microbiome. So these are just some of the things that some of you might want some general hints about. What can I do to improve my microbiome, gut-brain axis? Well, these are some of them. So thank you very much for your attention. Thank you very much for listening. This is the work of a great team of people. A big shout out to my now retired colleague, Ted Dinan and Jared Clark, and all of these wonderful people from all over the world who come to us in Cork in the south of Ireland to be on this journey, of really exciting journey. It's a journey that Hippocrates would have been proud of, because he also is thought to have said, let food be thy medicine. Now that could also be fake news, but I'm going to change that. Let food for your microbes be medicine for your mind. And sometimes, just sometimes, it's good to turn pictures upside down and we might see them in the right way around. Thank you very much, Sandy. Thank you for listening.
John, amazing. Oh my gosh. I think everyone wishes we had a whole nother hour to sit here and ask you questions. There's a number that have come in. I want to get to as many as we can in the next 15 minutes to see what we can ask. And, please continue to add those. Because we are in this time of stress with, so much actual crisis stacking, with the economic loneliness, the pandemic, health. What is it that you feel like from the microbiome? Do you feel like that it has gone down? And what is our hope of rebounding with that as approach to helping people to see a way through as we're coming out of this? For me, we think about brain health. I'm curious how we would start to put this in with, because stress and depression is so rampant now with everything going on.
Yeah, for sure. I mean, because we haven't been through a pandemic before, we have to go, so evidence-based on this is always challenging, but we know from other studies, and that's why we're doing these stress studies in the lab, and we have been doing them because we feel they give us some proxy about how individuals deal with stress. And so what we know is yes, the microbiome definitely, the more stressed we are, the worse it is for our microbiome. And what we're trying to encourage is, let's look at ways that are practical ways that individuals can actually, create resilience within this microbiome and prevent the volatility in the microbiome. And so that's why I wanted to focus at the end there about some of these things that we can do in your life that are just, because when we're stressed, we're more likely to eat junk food. Mm-hmm. That's part of, so if you can, try and increase fiber instead, would be a better way of trying to deal with that. Try and get diversity in what you eat leads to diversity in what your microbiome is. So fiber is king, and fermented foods are queen of this. And I really think that they're my things. If I tell people, so like, and I know fermented foods like are very things in certain cultures. In Eastern Europe, for example, there's a long history of fermented foods, but in Ireland we wouldn't have that. And so I telling people about this, they don't really. But you can make it more palatable. You can add it to a bit of yogurt. You can. I make kefir, which costs nothing. Because the other thing is we want to democratize this. People are going through hard times. They don't have money to go out to Whole Foods and get fancy solutions. So we want to come up with solutions that are actually there for the people who may benefit from it the most. And so things like fermented foods and fibers in vegetables and should be available to at most people's just right there for them. So I really would hope that that's one solution that we have. I think diet is really important in that way. Certain probiotics will benefit for sure, but we need more data and more evidence for certain strains because it's going to be very strain specific. And so, we need to kind of mind our microbes, is how I say it, and that will help us mind our brain. And I really hope we can. And I think that's, especially with the social side of this as well. I'm really, trying to get my kids to have these fermented foods, which is challenging, I have to say.
Yeah, that was fascinating to think about, that your microbiome can affect your social. Just some of our work, and I know almost 3 decades ago, I had a doctoral student want to look at the gut and brain for autism, and the fact that it's coming out is really interesting, and to see if we can come out of this. So I think all of you can see why we want Dr. Cryan to help us figure out in our longitudinal work as we can optimize our brain health across the lifespan, from teenagers that you started with to the end of life. Our microbiome is one of our greatest assets. So, John, one of the things that people are talking a lot about is gluten and how it causes problems. What is kind of the microbiome in gluten in terms of ideas that you see to research?
Yeah, so it's best studied in the context of celiac disease. So there's been a lot of
Okay.
work going on there in relation to celiac disease, and work from Elena Verdue's group, a really brilliant team in McMaster, are working on it in relation to there. It's not straightforward because it's the immune system gets in the way, and so teasing apart that triangle about the gluten immune microbiome interactions. So it's not something that we're really studying, and I don't see it as being a big aspect of stress. But we're learning a lot more about how diet is affecting the gut in general. The paper just out yesterday in Nature from Guy Buxting's group in Belgium, really showing in a nice translational way how certain antigens, including gluten, could be having direct immunogenic effects in the gut. And I think that's really exciting. But I think anything that we take in our diet has the potential to change our microbiome in certain ways. But fiber is my, fiber is my main thing that I'm trying to get people exercised about.
Yeah, and I agree with you. I mean, that's such an easy add and thing. So one of the questions that came in was, just back to diet, how much is, and the C-section, thinking that, that affected your microbiome, how much is our microbiome is in our control versus out of our control, that things happen and it's like, oh, wait, I am a C-section baby and this is what happens.
Yeah.
Or can we counteract that really is in our control? And so, yeah, just knowing that, but there's so much you can do and don't be stuck there. What would you say to in out of our control?
Absolutely. I mean, so much of this is one of the things, the messages that's one of the things I wanted to get home at the end there as well, is that your microbiome is modifiable. So your genetics you're stuck with. Your genetics you just can't do much with, but your microbiome you can. And so, so I think it's a good news story. And just because something happened in your childhood, and we're very interested in childhood trauma and childhood other, things that are quite distal, but there are different ways to solve these later on. And so you can target, so some of our studies really show that you can target the microbiome in adulthood and reverse some of the effects of things that happened in early life, especially a lot of our animal studies were showing that. So there's an awful lot of hope with the microbiome. And I feel, but the thing is, everyone's microbiome is somewhat different. So what works for you, Sandy, may not work for me. And understanding that is going to be an important part overall.
Right. So another question is, the medications for some of the psychiatric diseases, so let's just take depression, does it affect the microbiome?
Absolutely. We've shown this, particularly with antipsychotics more than antidepressants, but antidepressants do as well. And, almost a quarter of all medications in pharmacies affects the microbiome. And so what we need to do more and more is try and understand, okay, could that be contributing to the benefits or the side effects? And so some years ago, we showed that antipsychotics cause weight gain, and we showed that the weight gain in an animal model was driven by what was going on with the microbiome. So maybe, and this is maybe when we haven't got there, you could take, when you're on your medication, you could take a probiotic or a prebiotic, and you could counteract some of the side effects of without having any negative effects of what's going on, or you could augment the effects of, but them studies need to be done in humans yet, and I think it's something that we need to see a lot more of, but I'm quite excited about that.
Yeah, I think people would be curious. You've probably gotten everyone on that is listening to think about, hey, I want to know, what is my microbiome? Can you talk about what that means? Of if we were doing this in the Brain Health Project, what would these people be volunteering to do, and how would we, from a research perspective, see what their microbiome is to track over time how healthy you are?
So it's a field that's moving very fast, and so the sequencing technologies and the algorithms that are being developed are getting more and more sophisticated. So it's really exciting. But one of the things we're doing right now is by measuring the microbiome that's just present in the stool. So we take a stool sample and we look at the microbiome that's there, and from that we can infer what is the capacity of these bacteria to produce chemicals. So could they produce something like, for example, the neurotransmitter GABA or serotonin, or could they modulate tryptophan, or could they modulate bile acids or other things? And we can make these predictions just looking at the microbiome. And then you can say, okay, well, that might affect, if we have an upregulation of that, we can then see, okay, could that get to the brain? Could that, could we detect it in an imaging study? Could we see what's going on? And so you start to really develop, and so we have a lot of excitement right now about looking at these neuroactive modules and neuroactive potential of the microbiome, and really how it's shaping what's going on. And that's something that we're really excited about.
Yeah, wow. You've made me so excited about it. It seems like the microbiome, because it's affected by what we eat, if you take it in the morning versus night, seems like it would be very different. So can you talk to us about that?
Yeah, so
how much it changes?
Yeah, we definitely know there's a circadianicity in the microbiome, but and we can control this a lot in our animal studies. But in humans, we have to deal with that. That's part of what we have to deal with. And so, taking multiple samples will be the best way to do it. You can't get people to poop on demand, and so you have, it is one of the caveats that we have. But it's something that you would ask them, when do they pass the actual sample, and try and use that as whatever else. But there's lots of technical hurdles in this. A lot of the bacteria are aerobic. There are challenges, but there's an amazing project called the American Gut Project, which is looking at hundreds of thousands of microbiomes, and it's a kind of a crowdfunded based approach, and people can spend money on and get it done, getting their microbiome done. And they've used this then to look at large metadata about what's important, what confounders are, like things like alcohol intake will affect the microbiome. If you've had a really bad junk food, or a night with too much wine, these are all things that are going to impact what's going on. But if you have enough sample size, and if you've repeated measures in the same individual, I think having that's really longitudinal analysis is really powerful. You're seeing still papers published in Nature and Nature Medicine N of 2 because they're doing so many repeated samples. They're not common, but like they are still there. There was one in Cell with N of 7 a few months ago. So while like in the GWAS area, we need as many samples as possible, and we should learn from genetics about where we are. But when we look at individuals, we can follow them up over time. It can give you a lot of important information.
They talk about vagus nerve stimulation to help people and help some of the depression and things. Do you think that by looking at that it could disrupt the microbiome in good and bad ways?
Yeah, so we've studied the opposite. We've looked at vagotomy in some of our animals, and there is some work going on there. I think it's really intriguing. And the same with brain stimulation, other brain stimulations that would be used, for example, in Parkinson's, because we forget that it's a two-way direction. And we've been studying mindfulness and the microbiome, and we need to look a lot more at top-down regulation. So I view the vagus nerve stimulation is really interesting to me, because that's kind of what the microbes are doing. They're tickling the vagus, and so they're stimulating the vagus, and so they're getting to the brain through that way. So it's kind of like the convergence of mechanisms.
Wow. What would you say? So another question came in about Alzheimer's disease and the microbiome. Could that be an early indicator of detection or a contributor to making it worse?
It's amazing work being done now. Work originally from Switzerland, but also from Sam Sisodi's group in Chicago has shown that if you don't have microbes, you don't develop plaques in these transgenic mice, Alzheimer's mice. So there's a real excitement in the microbiome field in Alzheimer's. Also, microbes make certain amyloid is an antimicrobial peptide. So amyloid, which is what causes, so maybe it starts in the gut and travels to the brain by its actions on what's going on as an antimicrobial agent. Fascinating.
We have more questions than I've ever seen, almost 60 questions so we're not going to get to them all, but It sounds like your book. Is there a way we can plug your book that might have some of the answers that people
Yeah,
want?
the book is available on Amazon and all good bookstores. And it's by National Geographic Press. It's written with a really great journalist, Scott Anderson, and so it's written for the lay person to be able to get at this. So it's a good primer for people who may not have heard of the microbiome before, or this gut-brain axis. The whole goal was to get people excited about it.
Well, I love that you said chocolate is something that we can do.
Yeah.
to help with the microbiome.
Sure. Dark chocolate, though. Dark chocolate.
So, John, I don't know how you found time to be with us today with everything you're doing. Tell me that I'm glad that you're not jet lagging and traveling so much, and that your microbiome has probably gotten healthier.
It has. It's got healthier since we last met in person, for sure. A lot of it is about, I found for myself, is about taking control, and being able to create routine. And your microbes, because they really like routine, and I know what types of foods my microbes don't like. And getting Christmas behind me was good, because my microbes don't like the sweets and things so much, whatever. But our lifestyles we have, like I would be on a plane every week before the pandemic, so do change our perspective a lot on how we can actually navigate our microbiome. And now I have time to make fermented foods and things that I wouldn't have done before as much. So I'm like a born again fermenter.
I can't wait. I wish everyone on this call would send back to us 2 things that you took away that were like, wow, this is life-changing. I think that our diets will change just with this motivation. I wish we weren't out of time. John, let's make this year the year that we start our study on and adding microbiome and figure that out. And so with that, someone wants the name of your book.
Oh, it's the Psychobiotic Revolution. The Psychobiotic Revolution.
Psychobiotic Revolution. So, and we'll also send it out to everybody after this, because it sounds phenomenal. And I think you could say we definitely have a genius mind. So is it the brain or is it the gut? It's both, right?
It's both. It's both, but we sometimes have to turn them upside down.
Yes, I totally understand. So, Paige or Catherine, would you pull up the thing? So for you that want to be involved in making your brain healthier and your gut healthier, that can be involved in this is how you can sign up to scan this, the Brain Health Project, where we're going to be start enrolling 6,000 people this year and 12,000 next year. So we're looking for healthy people from ages 10 to 100 and follow them and make sure we do everything possible to make sure your best brain years are ahead of you and not get behind with all of these crises that we're facing. I also want to remind you that, here we want people to have access to the most breakthrough news in terms of what happens. So next week at Sips and Science, be sure to sign up. Kirk Erickson he's the exercise, and John, there actually were some questions coming in on how does exercise change your microbiome. So just but I want to plug his talk at 7:00 at night through Sips and Science, and then become citizen scientists with us here at the Center for Brain Health in the World and our collaborators, as we all figure out ways to keep our greatest asset and change our economy and make the world a better place. But just in closing, John, would you answer, since we want to kick off Kirk Erickson and exercise, tell us as a last kind of tease to hear him, how does exercise impact your microbiome?
Greatly and amazingly. And we're studying this now to see if the beneficial effects of exercise and depression could be mediated by the microbiome. And there was a study last year even showing that people who ran marathons, the New York Marathon, that when they transplanted that microbes to animals, the animals ran faster. So everything is in there. So it is, aerobic exercise in particular is really good for your microbiome diversity. So that's really exciting area.
Tremendous. Brilliant talk. Thank you for making time for us.
My pleasure.
Oh my gosh. We can't wait to bring you back here, but we're going to continue with these virtual meetings. Thanks for making
Oh,
time for us, John.
my pleasure. My pleasure.
Thank you. Terrific. Thank you all for being part of this, and we look forward to seeing you next week and other Fridays for our Frontiers of Brain Health to see the latest breaking brain health discoveries. Thank you.